Well, welcome everyone, and thank you very much for coming today to this information session on the SARRP, which is the Small Animal Radiation Research Platform that we've just acquired on this campus. And so what is that? It's an image-guided preclinical irradiator. And just to point out that this is the first SARRP in Australia, so it's really quite monumentous for us. And it's also the first image-guided preclinical irradiator on the eastern seaboard of Australia. So we're really very excited to have this, and it's really going to open up a lot more opportunities and collaborations for us on this campus. So I'm looking forward to exciting times with this instrument. And you're here to learn about it, but before we do that, I'd really just like to start with some acknowledgements, because for those of you who don't know, this has been quite a long and arduous task to actually get this far. So first of all, it all started with a 2016 Cancer Institute New South Wales grant that I put together, and I've listed here the co-CIs on this grant. So they were Zdenko Kunsik from CPC, Tom Eade from the Department of Radiation Oncology, Nick Pavlakis and Helen Wheeler from Medical Oncology, Alexander Engel from the Department of Surgery and Sydney Vital, Emily Colvin from the Bill Walsh Lab, the Kolling, Dale Bailey from Nuclear Imaging in the hospital, Will Stevenson from Haematology in the hospital, and Regina Bromley from the Department of Medical Physics in the hospital. So all of these people contributed their projects, their CVs to this grant, and without them it certainly would not have been successful. So I really need to acknowledge all of those people. We also, a prerequisite for this grant application was to have co-funding, and this was really initiated by Michael Back from the Department of Radiation Oncology. And without his say-so, we really weren't able to apply. We wouldn't have been able to apply for this, and we would have had to go for just the radiation cabinet. But ultimately, we had co-funding from Northern Sydney Local Health District and also from the DVCR at the University of Sydney. So all of those people and places are acknowledged for their contribution towards this piece of equipment, which is, has a list price of over $1.5 million Australian. So next, just getting the money, or getting the promise of the money was, I guess, the easy part. We had a lot of other work that needed to be done, and the procurement process was led by Lewis Nguyen, who is here today as well. And he really guided us through the tender process and did some amazing deals for us, which is why we have what we have today. But I was also very ably assisted in working out what we needed and what we should be getting and what we didn't need by Regina Bromley from the Department of Physics here, and also Nana Sun, who's in the audience from the preclinical imaging facility at University of Sydney. In terms of trying to navigate the funding situation or the finance situation in the middle of a review on this campus, I had amazing support from Jonathan Morris and Neil Asanga from the Kolling, and Vanessa Sanford from Sydney Vital also came in at the last minute and helped. And without all of their help, we really wouldn't have gotten through the procurement process. I'd also very much like to acknowledge Trend Bio, so Trent Warburton and Amarat Peterson, who were incredibly patient and worked with us, because our quote expired in March and we didn't actually say we'd get it until July, or it might have even been August. So they were incredibly patient. And finally, it's not enough to have a piece of equipment, especially something as whiz-bang as that. We really need to have a resident expert and a manager for the equipment. So my next step was trying to find funding to actually get a manager for the equipment, and I was able to get this from Sydney Vital and the Sydney Neuro-Oncology Group, and they've funded the manager, and we now have a very, very capable manager for this piece of equipment, Kelly McKelvey, who some of you should know. Her expertise is in vivo modelling and cell biology. And we're just assessing what sort of input we need in terms of medical physics and radiation oncology. And here we've got incredible support from the Department of Radiation Oncology to work out what's going to work best, so that all the preclinical work that we do with this instrument is going to be clinically relevant, and we can trust it in terms of QA and QC. So they're very much on board in working out what we need to be able to move forward with this. So again, I'd like to acknowledge Sydney Vital, Sydney Neuro-Oncology Group, and the Department of Radiation Oncology. And it's taken a long time, so I really have to show these photos. This is the piece of equipment arriving on our campus. It weighs 2.5 tonnes, and I think it took a bit to get it off the truck and installed. And there's a few more photos. We did have to go, the engineer Dan and I did have to go sort of dumpster diving into one of these to find some keys that had been misplaced, but we found those. I think the bolts are still missing, is that right? The bolts are still missing. And so this is what the basic piece of equipment looks like, but we do have an add-on for that. So we actually have this in our facility, so that's a huge landmark. And that's sort of where my role ends, and now I hand over to Kelly, who, as I said, is the SARRP manager, and she'll introduce our speaker for today. Kelly?
Okay, good afternoon. As Viva's introduced, my role is to be the SARRP Preclinical Irradiator Manager on the campus here at Kolling, and that's really to facilitate between the researchers and any assistance that we're going to need from Extral or from the medical physicists in terms of the radiation treatment planning. And that could be anything from helping with the animal ethics to giving you some guidance about what's clinically relevant for your radiation dosing, as well as maybe some of the beam arc therapy that needs to go ahead there. In the last 2 weeks, I've really had a great time spending a number of hours down in our facility with Wilfried and also with Daniel, who's our engineer, both with the installation and also with the training more recently. And Wilfried is incredibly personable. He's highly charismatic. He has a wonderful French accent, which you'll get to experience in a few minutes. So Wilfried comes to us. He's done a Master of Science and a Master of Business Administration in France originally. He's come all the way across the Pacific Ocean with Daniel from Georgia, Atlanta, leaving behind his wife and 3 children, probably quite happily. Before he went into science, he was working as an assistant director in one of the national parks over in France. This has a 300-hectare national park as well as a very large lake in the middle of it. So that's quite a step into science then when you think about it from that. So in 2003, he left France and joined the United States, where he's worked all the way up from a lab helper through to a research lead specialist, the Department of Radiation Oncology at the University of Maryland, where he spent 12 years working in adaptive immune responses. And this was really working with some really interesting animal models using xenopus, which is a type of frog, if you're not familiar, and also the nurse shark. It's a big step from our mice and rats that we now have in our SARRP preclinical irradiator. He's also spent 5 years assisting NASA's Space Radiation Laboratory to try and develop and assess the radioprotectors against the cosmic radiation, and also looking into DNA repair, training a number of NASA students in both the cell culture and also some of the irradiation techniques. So he has really a wide range of technical expertise, all the way from using a microbeam electron gun, which sounds very impressive to me, a cabinet X-ray, which is similar to what our SARRP is now, all the way through to what the clinical linear accelerators are. So the SARRP really sits between a traditional cabinet X-ray machine and the linear accelerator that we have over in our hospital next door. So 1 year ago, or just under 1 year ago now, Wilfried joined Xtrail as one of the experts and the application specialists for this piece of equipment, as well as a number of other of Xtrail's instruments. And it's really the sort of 2 years that he's worked hands-on with the SARRP to really give us as much information, as much insight from his own personal experience as he could. Taken a number of extra notes, which we've added onto the training manual. And it's all those little bits of insight that we really couldn't gain from a manual per se. And really enjoyed being able to spend that time with Wilfried, and also having Daniel over here giving us all of that incredible expertise that they have. And as Viva said, that by having this piece of equipment now in the Kolling Institute, we have a very distinct advantage in terms of our cancer radiation oncology research here in Australia. And also this brings, especially for the patients, because of course, while we're preclinical researchers, it's always the patients at the end of the day that we're really trying to help. This brings our research much closer to the clinical scenario, and very fortunate that we have a piece of equipment that's going to allow us to do that. So thank you, and I'd like to welcome Wilfried down to the stage, please. If you give him a round of applause.
Thank you. I have mine. Is it working? All right, all right. Yes, I did. There we go. Well, thank you so much, Kelly. I didn't remember having such an impressive bio, but it's great. I want to start to say, but if you don't understand me, it's totally normal. Okay, my accent is thick, I understand. Welcome to the SARRP launch event. My name is Wilfried Goetz. I'm the application specialist with Xtrail. Formerly, like Kelly said, I work at UMB, University of Maryland, Baltimore, for 12 years as a research lead specialist. And I was a SARRP expert user, if we can say so. SARRP stands for Small Animal Radiation Research Platform. We were using this instrument in a CRO fashion, preclinical contract research organization. Right. I'm going to be a little slow with this. So why are we here today? So I don't really know. We should stop right here and go to the pub maybe. Obviously you already acquired system because you understood the need for more accurate instrument. Cabinet X-ray are underperforming and are lacking accuracy. You've seen the need and the demand for translational modeling, quality over quantity, and being confident of your beam delivery. We can do better than regular cabinet X-ray. If you can't see it, you can't target it. The goal of a radiation oncologist is to deliver a dose of radiation to a specific area in the most conformal way. But basically, you want to be confident of where you are delivering, minimizing tissue toxicity as much as possible. Currently, clinical trials are failing. And why is this? Statistics are showing that there is a lack of translational data that has been developed in the preclinic. Maybe this is due to underperforming equipment, which aren't keeping pace with clinical practice. We cannot solve our problem with the same thinking we used when we created them. Also, we cannot keep generating data that aren't translatable to clinical practice. For example, this is used to be cutting edge in a clinic, an old Van de Graaff AP treatment with shielding. No imaging, just a kind of point and shoot method, similar to what we have in a regular cabinet X-ray. This one is the first patient being treated at Stanford for a retinoblastoma with a generation 1 linear accelerator. Same thing, no imaging, just a point and shoot. Another example of an archaic way of treating an epithelioma with the patient holding its own shielding during treatment. My point here is we don't treat our patient like this anymore. Clinical practice has made significant improvement. Cone beam CT guided linear accelerator, complex and precise treatment planning system like the Variant Eclipse, And now we're getting to the MR-guided radiotherapy for even increasing targeting accuracy. My words came out fine. They were processed incorrectly by your brain. It seems that the data we are developing in the preclinic isn't being translated because the clinical practice is so far ahead. All right. I'm missing something here. See? There we go. I think we've done a very good job with the animal model, as we have immunocompromised mice like NOD, SCID, all these different type of knockouts that can have human disease, different type of cancer, which is great, but our radiation modeling is still poor. So far we have only half of the translational secret figured out. You'd probably recognize those. This is still standard practice using a jig and a lead shield with cutout, like the orthotopic glioblastoma example on the right. The source most likely is a regular cabinet X-ray again, irradiating the tumor from the top down. Obviously, this is not how we would treat human patients nowadays. Okay? If you are currently using cabinet X-ray single plane fixed source in a small animal cancer model, then it's a dinosaur. Cell culture is fine, but animal model needs to be treated differently. Although I also want to point out that this doesn't mean that what we've done until now was not good research. It's probably time to level up to clinical practice though. In order to reproduce the clinical radiotherapy process, we have designed the SARRP as translational tool. Hence, its capabilities must mimic the clinical practice workflow and protocols. These are 4 common steps in clinical radiation therapy that we reproduce with SARRP. The cone beam CT imager, a capacity to fuse with other image modality like MRI, a TPS treatment planning system that can allow you to verify and deliver non-complanar arc beams like in the clinic. The answer to failure in translational is the lack of targeting. So what the SARRP brings is the CT modality, as well as 3-dimensional optical system named MurayGlow. Now let's take a look on the inside of the SARRP. It's fully shielded. It's going to any lab space. It has a 360-degree rotating gantry. It is currently equipped with an orthovoltage 225 kV X-ray tube. It has adjustable filtration, aluminum for imaging, and copper attenuation for beam hardening. A 20 by 25 centimeter high resolution CT panel detector, which allows you to image multiple mice or a rat. It has a portal camera for fluoroscopic live imaging, which is used to check alignment and do beam's eye view for quick, easy image. But you can also serve, that could also serve in the near future for portal dosimetry QA. The robotic stage moves in the X, Y, Z, and theta direction. The SARRP is on wheels and fits standard doorways, although we had to take the doors this week. So no need for knocking down walls and rebuild the facility around it. But you get my point. Just a technical defect. I'm not done. Huh? Here are some of the different beam arrangements you can achieve. Non-complanar, APPA or parallel pose, continuous arc, conical arc, static beams, and a TPS treatment planning system which allows you to do forward planning. There you go, a little video. What do I mean by non-complanar beam using a 360 degrees rotating gantry and rotary stage? Well, this video demonstrates exactly what SARRP can do. You can deliver a beam of radiation from basically any angle. Acquiring a CT takes only 67 seconds. The reconstruction being made at the same time, so that as soon as your CT is ending, it is already loaded in your TPS. The system is medical quality. Extral is both life science and medical device company. So we use the same components for both types of equipment, which assures reliability. The system is very accurate and allows for reproducible results. It's an open platform, meaning when we develop a new technology for it, they can always be adapted easily to the system that you already have. I have another video here. In this video, you will show what the SARRP looks like. You'll notice that leaded glass window in the front, allowing you to monitor your subject during treatment. 2 access doors on the side and 1 on the back for routing anesthesia lines, gas scavenging tubing, cable, etc. It rolls through your standard doorway, but you don't have the standard doorway here. The front openings are slim and convenient sliding bunker doors, so they don't take room in the space. There is also a webcam inside which allows you to monitor your subject from the control station if you don't want to go and look through the window. The TPS, treatment planning system, is composed of 7 easy steps. CT imaging, image registration, segmentation and contouring, dose planning, dose evaluation or verification, treatment delivery, and reporting. Another video. We acquire a CT through a pancake geometry, which means the detector and the X-ray tube are static and the animal moves in the theta circular direction during exposure. We found that we get better spectral resolution with this setup compared to a tubular geometry. Here's a picture of 3 mice laying down on a bed prior cone beam CT acquisition. As I mentioned it earlier, a CT acquisition takes only 67 seconds, and its volume reconstruction is being done real time. So what you obtain after it's done? This. Not this. This one. The reconstruction is loaded automatically into your TPS, showing 4 different views, axial, sagittal, coronal, and the 3 planes together. You can either go through the slice individually by scrolling with the mouse wheel or sliding the slicer above each view. You can adjust your window and level in order to change intensity and contrast. Then you can go to step number 2, which is registration. In this step, you'll have the possibility to co-register another CT, MRI, or basically any image modality or any volume with a DICOM format. You can also skip this step and go directly to the segmentation module. This is a really easy step where the TPS does everything for you. It will identify 5 different densities from the CT gray level: bone, tissue, fat, lung, and hair. The TPS will take in account those volumes with different density during beam calculation. You could also transform everything in tissue if you wanted to. You can save your plan at any point of your experiment and come back to it later, or load this plan for an ulterior occasion, a different mouse at 1 week, 2 weeks, 3 weeks from now. This is the contouring step. You can contour region of interest, but also region that you'd like to spare, which is very important. And this will be used during the verification step when you generate a DVH or dose volume histogram. Here we've contoured a lung using the automated level tracing tool, or you could also contour manually with the paint effect tool. When you're done with the contour, you click next and place your isocenter. An isocenter is a point of target for beam, usually the center of mass of your tumor or region of interest. You can add beams to this specific isocenter or to several isocenters. If needed, then compute those and go to the next step, which is verification before treatment. In this view, you can see all beam entry, isodoses, 3D dose volume, and a great DVH for target, since here, in this example, we have nearly the full dose, 100% of the dose, being delivered to 100% of your volume. At this point, if you're okay, which I would be with this plan, you could choose to save it and execute it. Lastly, you have the possibility to finish this procedure by generating a report. So So I told you a lot about the system itself. Now I would like to also show you some studies that were done using the SARRP. We have probably around 120 papers that have been published with the SARRP. I've selected only a few of them because I slacked a little bit in reading those publications lately, being very busy on the road. This is a good study that shows translatability in reproducing hippocampal sparing irradiation technique in mice. The study came out of Albert Einstein. It was published in Nature. The result mimicked an RTOG0933 trial where mice were getting whole brain irradiation as well as giving hippocampal sparing irradiation, which is easily feasible with SARRP, but not so easy with cabinet X-ray. If you don't avoid hippocampus, you can get neurocognitive dysfunction, a decrease in neurogenesis and cell proliferation. You also increase overall animal death. This study is of Beaumont, an early adopter of SARRP. What they did was basically compare what we've been talking about this entire time, cabinet X-ray versus SARRP. They had the standard model with intracranial glioblastoma using a lead jig. This is how you irradiate mice in a cabinet. You put them under azofluorine or a mix of ketamine xylazine. You nuke them from top down. It's really hard to avoid the optics or to spare the salivary gland, the trachea, the esophagus. But if you target the brain with a SARRP and you plant parallel opposed beam in a vertex field, you can easily spare all those organs. Therefore, you can give them a more clinically relevant dose as the subject will survive and won't die halfway through treatment. With a cabinet X-ray, you might achieve 30 gray, when with a SARRP, you'll go up to 60 gray. Beaumont had a MR and a PET system as well, so they were able to fuse or co-register this volume with the CT for even more accurate targeting. The findings are pretty cool. As you see, the control animal needed to be sacrificed really fast after only 21 days. With the single beam, they were able to get 2 gray per day for 5 consecutive days with 2-day break, but they had to be sacrificed after 27 days. Although with the SARRP, the tumor delay was much greater. The dose was clinically irrelevant and the survival was excellent compared to control and single beam model. Another big concern in a clinic or issue in the clinic when offering radiotherapy is to minimize normal tissue toxicity, especially in head and neck patients. Because of this concern, there are a lot of studies going on in the preclinic right now regarding radioprotectors and radiomitigators. What you can do with the SARRP is trying to replicate clinical problems, find a resolution, and move back into clinical practice. For instance, this study on the left from Beaumont, they were looking at radiation toxicity to the bladder. Radiation toxicity to the bladder is inevitable when irradiating females that have cervical cancer. In order to mimic this, they use a rat model sitting vertical, so its abdomen will drop, separate, and allow for increased targeting accuracy. Then they investigated radioprotectors to overcome normal tissue toxicity, looking at incontinence, bleeding, inflammation, and so on. They found that tacrolimus used as a radioprotector works for this model and was then adopted in clinic. On the right, a study came out of the University of Pennsylvania which looked at targeting a very small area of the intestinal tract, like a 5mm area. They surgically implanted a radiopaque marker and investigated curcumin as a radioprotector. This group found that indeed the area of the intestinal tract which were irradiated and treated with curcumin were showing increased radioprotection. Now curcumin can be used clinically as radioprotector as well. There's also a lot of buzz in the past few years regarding immunotherapy and cancer, and we look at 4 different papers from 4 labs, 3 separate models with different immune checkpoint inhibitors. And what we found is that if we add up all the animal survival amongst all the papers, then you find that in combination, targeting radiation with SARRP plus immune checkpoint increased tremendously survival. This also shows us how reproducible studies can be conducted with SARRP. Here it's finally where I introduce a little more my company. Extral is both medical device and life science company. We make orthovoltage cancer treatment system. We have a 100, 200, and 300 kV for non-melanoma skin cancer. We are also launching a new photoelectric therapy unit, which has an 80 kV X-ray tube, and it's used mainly for superficial treatment. We have about 700 medical systems worldwide, 70 cabinet irradiator, the single plane, which you shouldn't use for small animal cancer model anymore. And we have around 61 SARRP and 4 Xenex worldwide. Extral is all about customer support, and this is probably what sets us aside from other radiation-based companies. We employ experienced researchers that support our customers the best way possible by truly understanding their needs. We develop almost all our product in-house, but also we benefit of help by collaborating with prestigious groups such as Johns Hopkins, where the SARRP was first developed at. We use obviously phone, emails, computer, remote access software like TeamViewer, or on-site troubleshooting. Trend Bio, our partner in Australia, will also partake in supporting your needs. Yes? All right. All our systems are commissioned in-house. Actually, I take care personally of the commissioning. The SARRP is used at many levels: university, research, hospitals, big pharma, and contract research organization. Okay? Let's talk a little bit about the additional accessories we have available for the system. We have many different animal beds, 1 to 3 mouse bed, rat or mouse vertical bed, a universal bed that can go easily into a MR and goes back onto the SARRP, keeping the animal positioned for cleaner fusion. We have heated bed as well, so you can maintain the animal body temperature while under anesthesia. They can have integrated isoflurane mask, vertical option are also available. Lastly, we are going to talk about MiraGlow. You guys have MiraGlow here, which is very exciting, and also the fact that you are the first SARRP in Australia, and therefore you open a full new continent to us. I'm pretty sure most people here are aware of what bioluminescence is, but to simply describe it, I could say that you start by transfecting your cell with luciferase protein. This is a conserved gene. So what it means is that the cell will proliferate and multiply with that gene attached to it. You implant the cell or the tumor into the mouse, and then before you do imaging, you inject your subject with luciferin, which will bind to those sites. Then you expose your sample and collect images under a specific wavelength sets. I hope I'm not boring you to death right now. CBCT does not effectively differentiate between soft tissue and tumors. BLI has become a standard in tumor imaging for preclinical model. It is cheaper than PET and allows for quantitative analysis of tumor growth. Existing systems generate 2D bioluminescence imaging. This does not provide depth of the tumor. In irradiation studies, this can mean irradiation or radiation is being delivered off target if the tumor is deep within the tissue, like we have right there. As you can see here, the 2D BLI can be misleading, especially if you want to target your region of interest by just checking one plane, then you'll be missing the target depth accuracy. If you have two targets sitting close to each other, then it could be perceived as only one big tumor, when they are in fact two different regions. So we developed MiraGlow. It is a three-dimensional optical system with a rotating mirror assembly that captures light intensity from all the way around the animal. It has a high-resolution cool CCD camera, charge-coupled device camera, that records precisely the emitted light. It has reconstruction protocols and algorithms which convert the light intensity data into a center of mass that becomes your isocenter. The system may soon accept a fluorescent camera, and this is currently in development. This slide is just showing you the mirror assembly used to capture light from all angles. Here we have another little video. Okay. This is an old video of the Murray-Glow. The present hardware is a little different, but the principle remains the same. What hasn't changed is the mirror assembly and how the emitted light is captured within the animal. The beauty of Murray-Glow is that it can function as a standalone or can be used integrated. Mounted on an articulated arm which can easily dock inside the SARRP. The same bed will be used for both BLI and CT acquisition, so the animal position won't change, which is usually quite the challenge when you're trying to fuse two different image modalities together. This is a GIF image showing the docking process into the SARRP. It looks like it was done here, actually. Oh yeah, the only thing, we have a different front now, okay. The bed, like I said, are heated as well in the Murray-Glow. There are fiducial markers on top and the bottom for geometry calibration purpose of both the BLI and the CT acquisition. The workflow is basically you acquire a BLI, you calibrate, acquire a CT, fuse, mesh and data mapping, then creation of a center of mass, and you can irradiate. You're going to be able to identify a tumor that you may have not been able to see with just a CT alone. Remember earlier I showed you an image of a 2D system having trouble dissociating two tumors close to each other? Well, Murray-Glow can identify different tumors that are just 3 millimeters apart. Now what's next with Murray-Glow? Well, we are looking at new algorithms to enable tumor volume quantification by optical intensity, as well as integrating a fluorescent tomography module. So now we are back to where we started, or are we? We cannot solve our problem with the same thinking we used when we created them. Although after this presentation, I presume we can all agree that we have a better idea of what we should use as far as instrument in cancer research study. If we want to produce clinically relevant data, we need to integrate new technology in our experiment, especially if we are seeking a translation of those data into the clinic. Now you see it. Now you can target it. What we are looking as a company is trying to do better by taking user feedback and integrating new tools and ideas customers would like to see appear in our system. The main focus we are currently working on is first bettering our imaging modality, as it only will increase targeting accuracy. And second, we always look to improve the experimental workflow, so then we could irradiate more animals per day. This is it. Presentation is done. I would like to thank Dr. Vive Howell for the acquisition, or Viva I should say, for the acquisition of our instrument, as well for the warm welcome in the institution. Many thanks to Trend Bio, our distributor, for their tremendous help and support. A huge thank you to Kelly McKelvey for organizing and planning everything for us. That's pretty much. Thank you for listening. Thank you so much. Well, I know there's no question because they didn't understand my speech. All right, because basically my talk was saying let's not use cabinet X-ray anymore. But the fact and the matter, you can actually use the SARRP as well as a cabinet X-ray. I've done it with open field. You may have a little Hill effect, a little lack of uniformity. But I've used the SARRP to put cells on the bed at at the isocenter level or the axis level, 35 centimeter. And to increase the uniformity, I would do 2 beams, parallel opposed or APPA. It gave me great result, similar to the pentac I had when I was doing my clonogenic survival before. So you can absolutely do a cell irradiation with the SARP as well. You could do a whole body irradiation if you wanted to. There are some, you can adapt some collimator for that purpose. All right, so I personally worked with some geriatric mice which were a little obese, and I was able to, the X-ray equipment is strong enough that you can actually do your CT even into a rabbit if you wanted to. You could use actually the SARP for this purpose. As far as bioluminescence imaging, which is maybe what you refer to, I don't have the expertise, but I would presume last time I used BLI, we used some mice that were above 30 grams, so you can consider them already obese, and the emission was perfect. So this is totally possible. Yeah, for targeting. So yeah, obviously the deposition, the PDD, the percent depth dose will be different on a sample that is bigger. But like I said, a mouse that is 25 gram or 35 gram is no problem for that kind of energy. At 225 kV, you could go through 5 centimeter of, or even more volume. Like you could actually irradiate a tumor which would be located in the middle of a rabbit sample. No problem.
Thank you for the presentation and also for our opportunity for research.
Thank you.
So you talked about the clinical state of the art. You showed MRI guidance and accelerator, but the trial has the CT guidance. So I'm sure you thought about MRI guidance monitoring systems.
Yeah, we have many development. Right now, we would like to maybe introduce some micro-CT equivalent. I don't think we're going to be able to, in this cabinet, bring an MRI capability. Right now, the only thing we thought about was allowing the user to bring 3D volume that were acquired on clinical equipment such as MRI or PET-CT, or you could also use ultrasound and co-register your ultrasound with a CT acquired. As far as the cabinet itself, we are kind of limited because it's compact, but we can think of maybe adding an extra tube or two here and there. So it's in development. I don't know how long it's going to take us to develop that. MRI, I don't think it's probably far off to be inside that same cabinet right now.
What has been your success rate with this type of thing?
100%. No, I don't have the data here. I can only say what is my experience. I used the SARP for 2.5 years. I had study for brain tumor, prostate. If you define your experiment the right way, if you take time to generate maybe a dose curve or dose response, you'll have great results. But if you want to take shortcut and go and decipher what should be the dose from papers that you find in publication, that might be a little more difficult. I would recommend that, like in any study involving radiation, you do your dose response. Then your success rate is going to be tremendous. It's a good question. You're right, they all need to be adapted. But to tell you the truth, it's very easy. I made my own bed. I made my own actually I came up with the 3-mouse model. Extral at that time didn't have the material required for me to put 3 mouse. As long as you use a material that is X-ray transparent, you can use anything you want. You know, so it's very, very easy to adapt from an MRI to a CT. You know, and especially now with 3D printers, you can make anything in 1 hour time. You know? Thank you. Say again? Oh, all right, so maybe I didn't show that. So you have the gantry on that's the X-ray source, okay? That's usually sit on top. When you do imaging, the gantry can rotate minus 180 degrees or 180 degrees. When you do a CT imaging, the gantry is going to be facing the detector panel, which is in the left side of the instrument. And that's why I told you, instead of the tubular geometry acquisition that you would have normally in a clinic environment, we have a pancake geometry. So your tube is here, your detector panel is here, the mouse is here, and there's a rotary stage, theta stage, that rotates the sample here. That's what we found, that we had a better spectral resolution with this. Oh yeah, well, especially this is based on the algorithm that we have. We have a software running in the background that allows you to do the reconstruction lifetime. So you don't have to wait. The 67 seconds that it takes to do the CT image acquisition, it's also what it takes to do the reconstruction. We have like some very powerful GPU that we have 2 big GPU on our desktop that take care of this. You're welcome. Well, it depends what you... I will have to relate that to TrendBio because I don't know really the price, but it's a trade-off. It might be a little more expensive than a cabinet X-ray, but what you're trying to do here is to be more accurate, to be more clinically relevant. So, yeah, it might be